Lyon's Hypothesis. UGC NET Anthropology. Welcome to AnthroNet Academy, a study aligned with the UGC NET Anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in Anthropology, complete with author names and insights important for UGC NET exam. Dosage compensation and sex chromatin bodies. In man, it has been found that Y chromosomes are genetically inert in comparison to the X chromosomes and other chromosomes, and only a few genes are present in the human Y chromosome. From the above discussion on the chromosome numbers of male and female human, it appears that females contain a higher dose of functional gene-containing chromosome than males. Female chromosome numbers equals 44 plus XX and male chromosome number equals 44 plus XY. Geneticists have observed that in some case, female homozygous for the genes in the X chromosomes do not express a trait more markedly than do hemizygous males. So, it must be a mechanism of dosage compensation through which the effective dosage of genes of the two sexes is made equal or nearly so. This mechanism of compensating the differential doses of functional sex chromosomes in male and female human is affected by the inactivation of one X chromosome in the normal female. The genetically inactive X chromosome or condensed X chromosome is called heteropyknotic X chromosome or heterochromatin or sex chromatin body or Barr body, according to the name of the geneticist. L. Barr, who first observed it, or drumstick, according to the shape of the inactive X chromosome. Of the two X chromosomes in females, which X chromosome becomes inactive is a matter of chance, but it should be remembered that once an X chromosome has become inactivated, all cells arising from that cell will keep the same inactive X chromosome. In humans, inactive form of X chromosome as a Barr body have been observed by the 16th day of gestation. X chromosome inactivation occurs in human when two or more X chromosomes are present. What is the Lyon hypothesis? Definition. The Lyon hypothesis states that in female mammals, one of the two X chromosomes in each somatic cell is randomly inactivated during early embryonic development. This process is called X chromosome inactivation, XCI, and helps to equalize the dosage of X-linked genes between males, XY, and females, XX. Key terms. XIST gene produces a long non-coding RNA that coats the X chromosome from which it is transcribed, leading to inactivation. Barr body. The visible, condensed form of the inactivated X chromosome in interphase nuclei. Mechanism of X inactivation. Timing. Occurs during the early stages of embryonic development, around the blastocyst stage. Random inactivation. In each cell, either the maternal or paternal X chromosome is inactivated randomly. Epigenetic process. The inactivated X chromosome becomes highly condensed into a structure called a Barr body. Inactivation involves DNA methylation, histone modifications, expression of the XIST gene, X-inactive specific transcript, stable maintenance. Once an X chromosome is inactivated in a cell, all daughter cells inherit this inactivated state. Dosage compensation, or Lyon's hypothesis. The inactive X hypothesis, or the Lyon's hypothesis, or the dosage compensation is widely known from 1961, which states that only one of the two X chromosomes in the homogametic sex is functional while the other condenses and is inactivated. The X inactivated in some cells would be that from the father, in other cells it would be that from the mother. Hence, any tissue in the body of a woman would be a mosaic of cells which would show dominance of all genes having diffusible products, but would remain a fine-grained mosaic for other intracellular differences. Such a mosaic of cells might be difficult to demonstrate, particularly among rigid tissues, although cells which can be separated and cloned might show antigenic differences. This hypothesis has stimulated many new investigations, some of which are currently being completed. Objectives behind the proposition of Lyon's hypothesis. Lyon was impressed by 3 observations relating to X chromosome. 1. In normal mammalian females, one of the two X's is genetically inactive in the somatic cells, single active X hypothesis. 2. Inactivation is random, i.e., irrespective of paternal and maternal origin, random inactivation. 3. A. The inactivation occurs during early ontogeny, early ontogenic differentiation. B. The particular X, which has thus become inactivated, remains inactive in all the succeeding cell generation, fixed differentiation. Evidences in support of Lyon's hypothesis. For single active X hypothesis. 1. Bangham, 1958, and Russell, 1961. Notice that an XO mouse is normal and fertile female, indicating that the activity of the single X is sufficient for the normal development of this species. 2. McNeil, 1956, recorded the case of calico cat or tortoiseshell cat, usually a heterozygote female with black and yellow patches. Here, the dominant X-linked coat color gene producing yellow color becomes inactivated in some cells, whereas in others this mutant gene produces yellow color, thus causing a mosaic appearance. Exceptional male calico is XXY. 3. X-linked ocular albinism in female heterozygotes causes the mosaic pigmentation of retina showing one active and another inactive X. 4. The late replicating nature of sex chromatin by H3 TDR and very little or no RNA synthesized by Barr body in human body indicate the metabolic activity of one X chromosome. In individuals having XXXY or XXX polysomic conditions. 1. There are 2 late XS. 2. 2 sex chromatins and 3. An apparent inactivity of G6PD genes in all but one X chromosome. For early embryonic differentiation. 1. Graham, 1954, while surveying the sex chromatin of various species of mammalian females, showed the absence of this structure in early embryos. 2. Despite its absence during early ontogeny, Barr bodies are noticed in late embryos of female cat. 3. Sex chromatins are deleted not earlier than 5 days in rabbit blastocyst and 16 and 19 days in human and macaque embryos, respectively. For fixed differentiation. 1. G6PD, which is much common in Negroes and Mediterranean people, but is virtually absent in North American whites in deficient activity, is quantitated by its ability for reducing methemoglobin with the consequent destruction of glutathione. 2. The enzymatic activity is now very conveniently assessed by the action on the substrate, and also the detection is made by starch gel electrophoresis. 3. By cloning aggregation of cells presumably originating from a single progenitor cell, the cells from females, heterozygotes for G6PD activity and employing mostly electrophoretic technique. 2 cell populations, 1 enzymatically active and other deficient, were detected. Summary table. Feature. Lyon Hypothesis. Proposed by Mary Lyon, 1961. Applies to female mammals XX. Purpose. Dosage compensation of X-linked genes. Mechanism. Random inactivation of one X-chromosome. Key gene. XIST. Result. Barr body formation, genetic mosaicism. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.